All right, so today we're going to be taking data from this study published by Gevers et al. And we're going to be looking at microbiome data in patients with Crohn's disease versus healthy patients. And this is a treatment naive data set, meaning these patients were tested for their taxonomic abundance in their gut microbiome before they were giving any medication. If you go all the way down to the end of the article, you should see some supplementary material. And this XLSX file is the Excel file. So once you download this, you should get this Excel file. And as you can see in the bottom, there's multiple sheets here, but we're only going to be looking at the Table 2I key taxa sheet. Now, if you look into these columns, you have the sample column, the subject column, AB exposure diagnosis. So CD means Crohn's disease, not IBD means not inflammatory bowel diseases, meaning healthy individuals, sample location. And then these are all the different bacterial families in the gut microbiome of the patients. After you download this file, we're going to be starting our R script. So at the beginning of our R script, we have all these packages that we are going to be installing. We're going to be using ggplot2 for plotting and data visualization, readxl for reading data files, vegan for alpha and beta diversity functions like Shannon and Bray Curtis, dplyr for data manipulation and filtering functions, reshape2 for reshaping data, specifically for stacked bar plots, and then also fileseek. You want to load in all the packages. Every time you run your R script, you want to load in these packages, but this part only needs to be done once. I've already loaded in my packages over here. You want to set your working directory to the file path that also includes your Excel file. My Excel file, I renamed it dataset. So I'm gonna run this. And then over here, we are reading in the specific data set. So I'm just reading it in and putting it into a variable called metadata. Then after you read it in, we are only going to be importing the key taxa sheet like I've shown. You want to use this function called the read underscore Excel function. And then you want to specify which data set you're talking about. That's the metadata. And then which sheet you're extracting. So we are reading that into our variable called key taxa to keep it simple. And now I am going to be dividing the columns into non-taxa columns, which are metadata columns. These columns basically describe the sample. And then we are also going to be listing bacterial families. Now we're going to be building the fileseek component. So the first component we're going to be making is the OTU matrix. So when we start this off, I just gave it a variable name OTU matrix. We are going to be going into our key taxa, and then we're going to be selecting all the rows. But for the columns, we're only going to be getting the sample column and all the taxa columns. And let me run this real quick. As you can see, our metadata is loaded in, key taxa. So if you look here, this is the same thing as the Excel file. And then taxa columns, these are character vectors. When you do your OTU matrix, we got in the sample column and all the other bacterial family columns. We're going to be converting that into a data frame. You want to convert into a data frame when you are manipulating the columns and rows, but you want to convert it back into a matrix when we make our final fileseek object. And then we are setting the row names for the OTU matrix to the sample IDs, which is done by this line. And then when I run that line, now you can see the row names over here are all the sample names. But now we have a column that's called sample and all the row names. So to get rid of that, we are going to be running this line, which basically says to remove all the sample IDs. And now if you look here, all the sample IDs are gone. At the very end, we're converting this component of the fileseek, the OTU component, back into a matrix. So now we have to make our sample data, which is also another part of the fileseek component. So when I run that, I'm going to be taking same thing as the last one, but only the non-taxa columns. So basically all these columns that I specified in this variable above. So when I run that, see sample data, it's just these columns. And then same thing, I'm going to be changing the row names into sample IDs right there. And then I'm going to be getting rid of the sample column. Boom. There you go. So now in our sample data, we have our row names as sample IDs, and this is consistent with our OTU matrix because our phyloseq wants it like that. And then in the sample data, we have our metadata. In the OTU matrix, we have our taxonomic family data. So after that, your third component is your taxonomy family name. And so basically what this line is doing is making a column name family and getting all the column names from OTU matrix. So if I run that, you can see over here, these are all the families that we're going to be looking at from our data set. So in order to assemble the phyloseq object, we need to combine these three components as I labeled 1, 2, and 3. So you have to use the phyloseq function. And over here, we are specifying that the OTU table gonna be the OTU matrix and taxa our rows equals false because we are saying that our rows are samples and our columns are taxa. And then our sample data, we're gonna put in sample data underscore F, which is this over here. And our tax table is this variable over here, and we're converting this back into a matrix. So your sample data can stay as a data frame, but your OTU table and your tax table have to be matrices. And then over here, we are basically going into our sample data inside of the phyloseq object and looking at the diagnosis column. And we are basically going to be changing this into a factor, which basically stores as categorical variables like CD and not IBD. And this just makes it easier for plotting. Now that we have our phyloseq object, we are going to be plotting our alpha diversity plot. So first, what we're going to be doing is converting our raw data into normalized data. And that is done by this line over here. And we're going to be doing this by using this function called transform sample counts. So we're taking our raw phyloseq object and we're going to be applying this function to normalize it. Now, the reason why we want to do this is because raw abundance, meaning the raw sequence counts for each microbial taxon, could alter the analysis because different samples are sequenced at different depths. So if you don't normalize them, samples with more reads would artificially look more diverse. So this is a very important step. This part of the function, we're dividing we are dividing each taxon's abundance by the sample's total abundance. So right over here, x divided by the sum of x. And then we have a safety check over here, which says if the sum of x equals 0, then just return the vector. And this is basically meant so if the sample has 0 reads, then it returns a vector instead of dividing by 0, which will give you an error. So now we're taking the OTU normalized table from our phyloseq object norm over here, and we are converting it into a matrix. And the reason why we're doing this is to make it compatible with vegan, which is a library, but we're going to be using a function in that library later on. So now we're going to be using that diversity function, which is in the vegan package, to calculate the Shannon diversity index. And this is basically going to give one Shannon value per sample. So let me run this real quick. This is the OTU norm table. So these are the normalized values. And then let me run the Shannon diversity. And that's over here in the values. So we're also going to be extracting the sample data from the normalized phyloseq object and converting it into a data frame. And the reason why we're converting it into a data frame is to allow for subsetting. So now that we have the normalized OTU table and the normalized sample data, we are going to be creating our alpha diversity data frame. And our alpha diversity data frame is going to include the sample IDs right over here, is going to include the Shannon diversity values that we calculated in this line. It's going to include the diagnosis, so patients with CD or non-IBD healthy patients. And then it's also going to include the sample location. So if I run this, oh, I forgot to run this. We should have our alpha diversity data frame. Now, if you look in there, we see our sample. The row names are sample IDs, by the way. We see our sample column. We see our Shannon diversity column. We see our diagnosis column and our sample location column. And now it is time to plot our alpha diversity graph. So what I'm going to be doing in this line is making a character vector called locations. And this is basically going to have the three locations from our data set: stool, terminal ileum, and our rectum. And I'm doing this so when we go into this for loop, we go over each location once for each iteration. So the first time is going to go through stool, the second time terminal ileum, and then the third time is going to go through the rectum. And this is also useful because I want to make sure for each location I have one graph. So in the for loop, we're going to first set a variable called site. And what this variable is going to do is going to take the data frame, alpha diversity data frame, and it's going to basically filter it out for each iteration. So only the rows that have the sample location which matches the sample lock is going to remain in this iteration. And this tolover function, basically what it does is it gets rid of case sensitivity. So if the capitalization is off, that shouldn't affect your results. So like I said, the sample location over here, it should match the sample location for each iteration as it goes through. And that's what it's doing. It's like filtering it out to make sure that we have the correct sample location for each iteration so we can make three graphs. And what this line is saying is if there are zero samples in this location, so say it goes through this and there are no samples for stool, then it is going to skip the rest and go to the next iteration. And now we are here to make our plot. Let me just run this line real quick. So we have our locations. So I just gave it a name called new plot, and this is from ggplot, by the way. So in ggplot, obviously, this is the data from site. And then for the x-axis, we're going to be setting it to diagnosis. And for the y-axis, we're going to be setting it to Shannon diversity. We are going to be using the geom box plot, and this is going to give us a box plot, which is going to result in a group level distribution. And then the geom jitter, this is really important too, because this is going to add points on top of your box plot, which is going to resemble individual samples. And then this function, the labs function, that's just going to give it a title. I'm naming it Shannon diversity by diagnosis. And then I'm doing this over here. So each time it goes through an iteration, it adds the appropriate sample location to the title. And then the x-axis, I'm giving it a name. And then y-axis, I'm giving it a name. I am saving it as a PDF. And similarly to the title, I am changing the sample location name each time. So let me run this for loop real quick. I don't know, for some reason it's not showing on my RStudio, but if we go into my folders, okay, so my plots are saved here, the file path in my downloads to my projects, and we have three plots right over here. So Shannon diversity by diagnosis rectum, stool, and the terminal ileum. And the reason why we want to separate it by location is because we're looking at species richness and diversity in a location. A higher Shannon index basically indicates higher diversity and species richness. So when we look at these three plots, we can see that across all the plots, the Crohn's disease patients have a higher mean Shannon index compared to the non-IBD. So that can suggest that patients with Crohn's disease have a higher microbiome diversity than non-IBD, meaning healthy patients. But the thing is, this cannot be a concrete conclusion because we haven't ran any statistical significance analysis. So if you're following along and you're interested in it, I recommend you to go look into it further and see if there is statistical significance. But those are the three plots that are produced by the alpha diversity function. All right, so now we're going to be plotting our beta diversity. And It looks very similar to our alpha diversity because it is at the beginning. We're going to be doing the same thing. We're going to be going through a for loop and we're going to be using the same variable sample location and iterating through locations, which are all the locations in our data set. These lines look identical to the alpha diversity, so I'm not going to go over them again. So now when we come to line 128, what we're basically doing is getting the sample IDs from site two. So we're getting all the sample IDs that are in this iteration. So if this is the first iteration of the loop, we're getting all the sample IDs for stool. And we're basically doing this to filter the OTU table, which is done in the next line. So we do OTU subset, and then we filter the OTU normalized table to keep only rows that match the sample IDs for this loop's iteration. So basically what we're doing is keeping all the rows in our OTU normalized table that match the sample IDs for each iteration and putting it into this called OTU subset. So for me to show you these lines, I have to run the for loop first. Site 2 just looks like this. And also, the reason why all these sample locations are rectum here is because I already went through the for loop. So the last location is rectum. So that's why site 2, you can see that all these are rectums. So now that we have our OTU subset, we are going to be calculating a distance matrix using the OTU subset. And to do this, we're going to be using a function called vegdist, and we're going to be using the method called Bray. And so what this is basically going to be doing is calculating the Bray-Curtis dissimilarity. And this allows us to determine how distinct each sample is within the exact location by comparing each sample to every other sample. And now in line 137, we're going to be using this function called CMD scale. And what this is going to do is perform a multi-dimensional scale using our calculated Bray-Curtis dissimilarity. And essentially what this is going to be doing is calculating the distances between samples. And this k equal 2 is going to be forming a two-dimensional map and it's going to be returning eigenvalues. And this is the equals true. So what this ORD is now is a list that contains coordinates of each sample in the PCOA space along with their corresponding eigenvalues. So now we're going to be making a data frame called beta underscore df. And for this data frame, the row names are going to be the sample IDs. And then the x-axis is going to be considered the PCOA1, while the y-axis is considered PCOA2. So when we look into this data frame, we can see how it looks like the sample IDs, the row names, and then the x-axis and the y-axis. Now, when I make the actual graph, I'll kind of explain a little bit more on what the x-axis and the y-axis mean. So now we're going to be merging this data frame because this data frame only contains these three columns, but we want a little bit more of the metadata. So we're going to be merging it with site 2, and we're going to be merging sample, diagnosis, sample location by sample. So if you look at beta data frame 2, we can see it has the same x-axis, y-axis, sample, but now it also has diagnosis and sample location. So then after that, we normalize the eigenvalues in this line. So we divide the vector of eigenvalues by the sum. And then what this will do is give us percent variation for our axes in the plot. So now we're ready to make the plot. I named it PCOA. And obviously we're going to be using this plot from ggplot2 or making this plot from ggplot2. For the data, we're going to be using our merge data, beta data frame 2. So for the X, we're going to be using the X-axis. For Y, we're going to be using the Y-axis. And we're going to be coloring by diagnosis. And we're going to be using geomplot for a point plot, and then we're going to be labeling the title using the labs function. Similarly to the last plot, for each iteration, it's going to produce one graph based on the location. So It's going to change depending on the iteration. And then for the x-axis, we're going to be naming it PCOA1 with this percent variation that we calculated over here. And the same thing for the y-axis. And then we're going to be saving it as a PDF. So now when I go over here, these are the three plots that we have produced. Now, when we look at these plots, what these axes mean, the PCOA1 and the PCOA2, what they actually mean is the PCOA1 axis represents the largest amount of variation in the microbiome composition. And the PCOA2 represents the second largest amount of variation. So what these percentages mean is basically 60% of all the differences between samples can be explained by how they are spread along this axis. And same thing for here, 20% can be explained how they're spread along the y-axis. So in total, if you add 60 plus 20, that equals 80%. So this graph is basically showing us 80% of differences that can be explained. And the reason the other 20% are not here is because they can't be explained by this 2D plot. So basically the way to read this plot is looking at each of these samples. So each dot represents a sample. Now, if the dots are closer together, that means that the microbial communities are similar between the two groups. If they're further apart, that means they're dissimilar. And we can see from this plot, I'll go over the rectum one first, they basically overlap. Like, yes, you do have a couple that don't overlap exactly, but it's not to say that you see any clustering, meaning, say we had a lot of red plots over here and a lot of blue plots over here, that would be considered clustering, but they're basically scattered around evenly. Some of these blue plots come out over here, some of the red ones come out over here, but majority of them are here. For stool, we see a little bit less of overlapping. We see more of these red points coming out over here, but still generally this isn't considered clustering because the red points aren't like really close together, and some of these blue plot points do come out. And for the last location, we see the same thing. So by looking at these plots, we can suggest that the microbial communities between the Crohn disease group and the healthy groups are pretty similar. They're not that different. However, like I mentioned, this cannot be an actual conclusion because we have not performed statistical significance analysis on it. For the last part, we're going to be looking at the taxa abundance composition between the two groups. So first, what we're going to be doing is getting the OTU table from our normalized phyloseq object and turning it into a data frame and naming it OTU DF. That's over here. And then we are going to be adding a row called sample. And if I do that, you can see the end of this, we have a row called sample with all the sample IDs. And I'm going to be doing the same for sample data. So we are going to be extracting sample data from our normalized phyloseq object, and we are going to be turning it into a data frame. And we are also going to be adding a row called sample with all the sample IDs. So as you can see, this is a new sample data F2, and this has the same row as the OTU-DF sample that we added, and this contains these columns. And the reason why we added the sample columns is so it's easier to merge them, because when you want to merge two data frames, you need to have a similar column. So now we are going to be merging our OTU-DF and our sample data frame. And we're only going to be selecting these columns from our sample data frame. And we are going to be merging it by sample. So after I ran that, if you look at this, we have the sample column that we merged it by. We have all of the bacterial families from the OTU-DF, and then we have the diagnosis, sample location from our sample data. So in the next line, we are going to be using this function called the MELT function. And what this is going to be doing is basically reshaping the data, and this just makes it easier for plotting. So if you look at the OTU fix data frame, you can see how there's the sample diagnosis, sample location, taxa, and then we have abundance. And this abundance basically is the abundance of each taxa for each sample. So if you can see, we have 13,520 entries. We have 13 total bacterial families in our data set. And we have around, let me go back earlier to our key taxa. So we have 1,040 entries, meaning 1,040 samples. So if you were trying to get the abundance of each taxa per sample, that basically means we should have the number of samples multiplied by 13, which is 13,520. So basically what I'm trying to say is we're calculating the abundance of each bacterial family per sample. So now in this line, basically what we're doing is we're taking the OTU function, and this is kind of similar to like piping in Unix. We use this syntax, and we are going to be taking that, and then we're going to be using this group by function. And what this group by function is going to be doing is it's going to organize the data by grouping them into diagnosis and taxa. And then we're going to take that, and then we are going to be using the summarize function. So after I run that, it should look like this. So we have 13 bacterial families, like I mentioned. So these are all the 13 families for the Crohn's disease patients, and these are the 13 families for the healthy patients. So these are the calculated mean abundance for each taxa per diagnosis group. And then we are going to be plotting our stacked bar plot. So we are going to be inputting our data as the taxa per diagnosis data. Our x-axis is going to be diagnosis, our y-axis is going to be mean of abundance, and we're going to fill by taxa. And this is going to be a bar plot. So after I run this, I'm going to save it. This is how the plot looks. So what this plot is basically showing is the mean abundance of each taxa in Crohn's disease and not IBD. And you can make comparisons to see, which group has more of what taxa, which group has less of what taxa. Now, like I also mentioned, none of this is statistically significant, so we can't draw any firm conclusions. However, it's still good to look at for further research. So from here, we can see the pink, um, which is Bacteroideales. It's a little higher in non-IBD and lower in CD patients. The blue, which I believe is the microcae, is also a little different between the groups. And also this color, which is, I believe, Clostridioides, is a little different. So that's basically the three types of graphs that I have generated. We have generated an alpha diversity plot using Shannon index. We have generated beta diversity plots using PCOA Bray-Curtis. And we have generated a taxa abundance plot using a stacked bar plot. And then this is basically going to help us understand further on how microbiome communities differ in healthy versus diseased individuals. And by understanding this and learning the differences, we can hopefully come out with cures, medications, antibiotics, or figure out how these communities change over time or what causes their changes to prevent people from experiencing these diseases and illnesses. So that is going to be the end of my video tutorial. I hope you guys enjoyed. I hope you guys learned how to plot these plots. And like I said, if you guys want to run your own statistical significance analysis, please do so. Leave a comment down below what you guys have found and let me know, if there is any statistical significance or if there isn't. And yeah, that's it. So have a great day, everyone.